We left off last time talking about resource ratio theory, and this is just two theoretical species. Species one grows quite well at high silicon, and this is the Monod equation, right? And species two doesn't grow as well, but species two competes better at high phosphorus and not as good at low phosphorus. This is another example of trade-offs. We've seen a number of trade-offs, and we'll see them, throughout the class, because that's sort of the way, one of the constraints of evolution. And you can think of it, either you can, have a cell membranes that have a whole lot of uptake proteins for silicate, or if you don't need to put all your energy into taking it up and getting scarce silicate, you can grow faster from other things. You're not limited by that, but you can use more silicate and grow faster overall. So this could be a smaller cell, for example, higher surface area volume ratio, or one that has less demand, diatom that has less silicon in its frustule than one that doesn't, or something like that. So what we also said is that the ratio of these things matter. That was the really important insight that Dave Tillman had when he went through this with diatoms, and he used diatoms that he grew from the Great Lakes to show that this matched reality, that the good phosphorus and the good silicon competitors could coexist. So if we have the concentration of silicate increasing on this axis, on the x-axis, on the y-axis we have the concentration of phosphate increasing. And there's what they call zero growth isoclines, and that if there's some level of silicate that's too low, species two is not going to be able to survive below that concentration, for example. And species one is not going to be able to survive below that concentration. And likewise with phosphorus. So species two can deal with lower phosphorus concentrations. Species one can deal with, lower silicate concentrations. Species one can deal with lower phosphate concentrations. If you have low silicon and high phosphorus, then species two wins because it's better competitor at low silicon and high phosphorus. Species one was a better competitor at high silicon and low phosphorus. So there's these zones where one or the other species is going to dominate. And this definitely works in chemostats, like a controlled environment with flow through, where you just keep the nutrients really constant at some level and you can mess with the ratios. But then, like I said, the same species can coexist in the field, and you can analyze the nutrients there and get a rough idea. And that coexistence is when the resource ratios are right here, sort of in the middle, right? Either both high or both low. Silicate and phosphate in this particular example. This is an additional explanation of Hutchinson's paradox of the plankton. Because if you remember, it said that all species have roughly the same requirements, and that's the part where that's not true. Probably the species that does better at low silicate doesn't require as much, right? You can say that, you can see that on this particular graph. But the rest of what the single nutrient is limiting, that's not necessarily true, because both species have different competitive abilities for different nutrients. But in a well-mixed environment at equilibrium, right? So a lot of our examples for counterexamples that might increase diversity of phytoplankton in lakes where there's not many nutrients around were based on this view that nutrients might be pulsing, either the zooplankton excretions pushing out pulses, or the rivers pushing in variable amounts of nutrients, or seiches cause entrainment, and there's mixing of nutrients in different weather conditions. Any of those things are all sort of non-equilibrium arguments. But even under equilibrium conditions, under a chemostat, which is a completely equilibrium system where inputs and outputs are balanced and everything's constant over time, you can get this coexistence. So it's an additional explanation. And you can think about this in terms of the fact that there's nitrate and ammonium and sulfur and all kinds of things that species need, potassium, iron, molybdenum, some of them. And each of those might have its own axis, if you think about that. I mean, you can think of a third axis coming out this way towards us out of the screen, right, for nitrogen, or like ammonium or something like that. And then you can get 3 or 4 species, right? And you can get 3 species in there, and then you have another axis, and so you can't really get around your head around multidimensional space like that, but you can get the idea that you keep adding species in with different, as you have more and more nutrients that are potentially limiting. All right. And another thing we can think about species interactions is in succession. And we'll get back to succession, but I wanted to talk about it here in particular with regard to biofilms and microbes, and bring it in the first time. If you have a copy of the book, you'll notice that there is an error. Prostrate means close to the surface, but your copy of the book left out the R. And so don't, somebody already caught that error. And I just won't say anything else about that. I'm sorry about that. Maybe it's an old man's mistake. So early on in succession, you have these scraping and rasping grazers that are able to, and that have to scoop stuff really closely off, and everything's tightly oppressed to the surface. So this could be right after a flood or in an area that has a lot of disturbance, right? But over time, we get these competitors, and so this might be like Cimbella, a diatom that's on a stalk, and if you get a bunch of those, then it's going to outcompete this stuff down here start outcompeting for nutrients and light, right? And then as you build the biofilm, a lot of these things make a mucilage or a mucopolysaccharide, and to hold themselves down on, and you get this sort of dense, gooey mat, which I'm sure, this is why when you step on a rock in a stream, it can be quite slippery. And so things start growing up through this, and eventually these large attached filaments are the ones that are going to take off over time. They're going to take over the assemblage. And in this case, we actually have more surface area for these small things that are attached to solid surfaces. They'll actually attach on to those filamentous algae with epiphytes. So this might be something like Cladophora, which we've seen in class and collected. So the point here is that the increasing light and nutrient competition is going from left to right. And that's so that the species interactions are becoming, these species are not so likely to be limited, these species are. And then the interaction with the organisms that feed on it. So we have the scrapers and raspers, then the collectors, invertebrates can come in here, gathering and shredding, and then piercing. There are actually piercers that pierce the Cladophora and suck out the cell contents. And then if you can be a scraper, you can get on the surface and scrape those epiphytes. And there have been some studies that kind of showed that when it gets like up to this really high level, the scrapers down here just can't deal with it anymore. So snails and things like that can't deal with it anymore, and they just kind of get stuck, and they can't really control it. But eventually, maybe you'll have a flood. Maybe you'll have a die-off of these. Maybe a virus will come in and infect them or something like that. Eventually, these things will slough off, and you'll go back to the beginning of the successional sequence. All right? And we'll talk about this in more detail, but there's some ideas like R species and K species, ones that grow quickly and colonize versus those that grow more slowly and are better competitors. Then we'll talk about mutualism, because we got to go through all the different interactions. One point is that highly co-evolved mutualisms are relatively rare in freshwaters relative to marine. So some of the really spectacular examples of mutualism are like, found on coral reefs, for example, where you have cleaner fishes and corals themselves have endosymbiotic, or animals with endosymbiotic algae, and some really spectacular examples, but they do occur in freshwaters, and there are some, and we'll talk about them here, and we'll talk about them again in the chapter on larger organisms and interactions that they have. There are N-fixing symbionts, which we'll talk about a couple examples in a second. So the ability to fix nitrogen is one that we know, like for example, legumes have bacteria in them that fix nitrogen. So there's a trade-off there that they can give each other stuff that's cheap. One of the ones that's really important globally is called syntrophy. And syntrophy is the fact that no organism can completely, when you go anoxic, can completely ferment everything down to CO2 and methane on its own. And part of the reason for that is hydrogen builds up. And you'd have to go back to the carbon cycle and that redox sequence. But when we get down to the bottom of that, the lowest redox stuff, there have to be multiple organisms. And so they essentially feed off of each other. Syn being together and troph eating together, right? And that just happens all over the world. And in a more diffuse sense, you can think of all nutrient cycles as being a mutualism, right? Because if one organism doesn't process and it can't take up a nutrient, then everything's going to build up in that form. If nobody could use ammonium, then all the nitrogen in the world would be ammonium, right? And so the organisms that use ammonium take it up, make it into organic materials, and we depend upon primary producers, right? Because we can't make amino acids or any of those kinds of things. So there's this mutualism between animals and producers and all and that. You can think of it in a way as, in a big sense, as being a mutualism. And I'll talk about the nostoc midge example because I think it's cool, and even though it's not necessarily microorganisms. And I'll get to this next slide has a series of different mutualisms that I'm going to talk about, and then also the background I can talk about a little bit here. The first one is Azolla, and it has Anabina or nostoc. It's actually taxonomically close to each other, and there's a little bit of disagreement over it. And Azolla is a water fern, and it photosynthesizes, but it holds cyanobacteria inside of these little pouches inside of it. And usually a cyanobacterium, anybody remember what this cell, specialized cell is and what it does? So cyanobacteria, specialized cell, no chlorophyll in it, yellow, yeah, fixes nitrogen, heterocyst, heterocyte, right, exactly. So the heterocytes. And what the Azolla does is that basically makes these things really nutrient limited, and it pushes them to have a whole, mostly heterocysts instead of one every 10 or 15 cells. You can see they're back to back basically a lot of times. So this thing is fixing nitrogen for it and providing protection from grazing. Really important in terms of the history of civilization, because before we had the Haber-Bosch process, which is the chemical process we use to fix nitrogen, right? There was no real reliable source of nitrogen fertilizer. And the traditional methods of rice culture in Asia, Southeast Asia, would be to flood the field, and then this Azolla would cover the entire thing, and with the nitrogen fixation, dry the field down, the Azolla will die and the cyanobacteria will die and release the nitrogen into the sediment, and then reflood it and plant the rice. And you could do that again and again, even though there's nitrogen losses from denitrification, you can bring it back in from nitrogen fixation and can repeatedly crop the same wetlandy field, which they built the entire civilizations in India and China and other parts of Southeast Asia on being able to provide rice for people to eat year after year after year. Hydra is another example. The Cnidarian that puts green algae inside of it, and if you grow them in the dark for a while, they'll get rid of the green algae. But in the light, they use photosynthetic organisms. I always thought that'd be kind of fun to have algae growing in me that I could go out in the sun and not have to eat. But I don't think there's many animals that our size that are able to live that way. But microbes are able to do that. This next example here is epithemia, and it's a diatom that grows in nitrogen limited situations. And if you look really carefully down in here, you can see these little blue-green bobs. Those are actually cyanobacteria, single-celled cyanobacteria. They're able to fix nitrogen. So it's the same thing. They provide the photosynthesis and protection from grazers, and inside of this nice silicate shell, the frustule, And the cyanobacteria fixes nitrogen for the epithemia. And these are widespread in nitrogen-limited streams. There's a similar mutualism to this that happens in marine diatoms as well. So it can be globally important, actually. And then on the right is a heterocyst, and this is an electron micrograph of a heterocyst with bacteria on the outside of it. And remember, from when we talked about nitrogen fixation, they have to protect the nitrogenase from oxygen. And what they'll do is they'll pump organic carbon out into these bacteria. Then the bacteria respire the oxygen away. So they actually, it's a mutualism. And if you cure them of the bacteria, they're not able to fix nitrogen as effectively and grow as effectively. So we've done these experiments to do that, to show that. The next example I'll talk about is the image behind me, and this is the surface here of a cyanobacterial colony, and there's a alga, a diatom growing on the surface of it. Remember that cyanobacteria have slightly different photosynthetic pigments. They have phycobilins, and that actually gives this brick red sort of color to the fluorescence of this surface of the cyanobacterium. And the chlorophyll A and B in the diatoms has a brighter red color, so you can distinguish between those. The reason that there's a mutualism involved here is that there's a snail that lives on the surface of this nostoc and scrapes the algae off of the microscopic algae off the surface of the nostoc, but it can't eat the nostoc. So it allows the nostoc to grow and keeps it from being grown over by the epiphytes. And the last example is the nostoc and midge one. And this one's cool because nostoc is a prokaryotic nitrogen-fixing cyanobacteria. Bacterium, not really prokaryote, that's old terminology. But the midge larvae lives inside of this nostoc and until it emerges. And right before it emerges, it somehow manipulates the nostoc to get all mushy so it can emerge out of there, and then the nostoc is fragmenting and making a bunch of new nostoc in the stream, and then the midge mates, Crocodopus nostocacola mates, goes down, lays their eggs, and the new midge larvae have new colonies to get into. If the midge is not here, the colony would be a sphere, but it's actually an ear shape because of what somehow the midge is manipulating by eating the inside of it. And they have silk, and they attach the colonies onto the rocks. So if there's a flood, these things are much less likely to get washed away than the nostoc colonies that don't have it in there. Only one nostoc midge per colony, and if I did the experiments as a graduate student or a postdoc and threw 2 midges in here, and they fight to the death. You know, somebody wins and the other one doesn't do so well. If you take the midge out of the nostoc, it's sort of a similar thing to this nostoc, other nostoc species. The midges actually stick their heads out of a hole and kind of clean off the surface of this nostoc. They eat the nostoc itself, but they also clean all the diatoms off of it, so they keep it going. These midges larvae are horrible swimmers, and so the fish would just cream them basically, or predatory invertebrates would just cream them. So they have a really nice, dense, leathery home that they get. They help the nostoc reproduce, and in return they get food and shelter. So it's an interesting one. The reason that I think is sort of one of the most interesting ones is that you have a eukaryote living inside of a bacterium, right? So it's completely flipped. Every other symbiosis, the microbe lives inside of the eukaryote, right? Every single one of these, it's the microbes inside, rhizobia, even the terrestrial ones. So I think that's kind of a cool one. And the last topic is the idea of chemical mediation of microbial interactions. And most microbial interactions are chemical, with each other. So we have these biofilms, right? And it's either competition for nutrients, and it's because they're not big enough to really, most of them are not big enough to sense light. You have to be able to be a certain size to basically get wavelengths of light that you can see. And all organisms use chemical cues that we know of one way or the other. But microbes are really, mostly that's how they interact. We do need to consider hydrodynamics, such as Reynolds number. So, for example, if you are in an open water, you can have diffusion much quicker in and out, right, of chemicals. And you don't want to put out a chemical that's costly to alter the behavior of your neighbor if you are in a place where you've got high exchange. But if you're down in a biofilm where the molecular diffusion predominates and everybody's close together, then you want to do that. So there's evolutionary constraints depending upon the habitat the organism's in, and it allows that allow organisms or constrain the way organisms are evolved to interact with each other. So for example, in a stream, if you're in flowing water, what you do here is going to influence who's downstream, right? But what this one does down here isn't going to influence what's upstream. So there's really no sense in like putting out a chemical to stop competition or something like